Today we'll be talking about the DNA structure and forces surrounding it. So DNA is a double helix structure that is anti-parallel and is made of nucleotides. Here's a basic structure of the nucleotides. There are three basic components to the structure: phosphate group, sugar group, and nitrogenous base. Again, this is a very simplified version of the nucleotide. Let's start with the phosphate group. This is the molecule that will link each of the nucleotides and create a chain that we'll see later. In the sugar group, you will see two molecules. The one on top is a ribose sugar, which is found in RNA. The one on bottom is a deoxyribose sugar, which is found in DNA. In the nitrogenous bases, there are two groups. Purines consist of adenine and guanine, which are double ring structures. Pyrimidines consist of thymine, cytosine, and uracil. Uracil is found in RNA. Notice that pyrimidines are all single ring structures. Now let's go over the bonding between these two nitrogenous bases. Adenine always pairs with thymine, guanine always pairs with cytosine. Hydrogen bonds are what keeps the bases together. These are pretty weak bonds and do not contribute to the overall stabilization of the DNA. Now let's take a closer look at the sugar groups and phosphate groups. Here you can see that there is phosphodiester linkage bonds, which connects the 5' phosphate to the 3' OH group. The sugar group's carbons are numbered in a certain way, which translates to the alignment. Let me explain. Here we can see that it is labeled 1, 2, 3, 4, 5. The first carbon attaches to the base. The second carbon is where the OH would normally be on the RNA. The third carbon is where this connects to the next phosphate group. And the fifth is where the previous phosphate group attaches. One of the important properties of DNA is also shown here, which is how it's anti-parallel. We can see that the left-hand side has a 5' end on the top and 3' end on the bottom. On the other side, the 5' end is on the bottom and the 3' end is on the top. This is due to the flipped appearance of the sugar group on the right-hand side. The next major concept is the major destabilizing force in DNA. We know the hydrogen bonds are weak, so what keeps DNA together? It is the van der Waals forces between bases contributing to base stacking. Rather than the horizontal bonds, it is more so the vertical attraction that keeps the DNA together. The minor groove is when the DNA backbone is closer together, whereas the major groove is when the DNA backbone is further apart. Now let's talk about handedness. This refers to DNA coils. As you can see, if the coils are going upward and to the left, like here, then that is called left-handedness. If the coils are going upward and to the right, then that is called right-handedness. Lastly, the negative charges on DNA backbone are destabilizing. When placed in a salt solution, the charges are neutralized. Once the charges are neutralized, the DNA has less of a destabilizing effect. It is stronger and needs more heat to denature. Tm is the temperature where 50% of the DNA is denatured. So when placed in a salt solution, the DNA would require a higher Tm to denature, as shown in the chart provided. Here we can see how the Na here, the salt, is attaching to the phosphate groups, which are negative, and that is how the charges get neutralized. Here are my references. Thank you.